And we pick up our lecture on Chapter 1 with our third way to collect data, an experiment. An experiment deliberately imposes some treatment on individuals in order to observe their responses. So an experiment is different from an observational study in that you actually impose treatments. You might randomly allocate subjects to 2 or 3 different groups and assign different dosages of a drug, for example. Since you are applying treatments, you are disturbing Mother Nature. You're doing something different than what would have naturally occurred. The whole purpose of an experiment is to hopefully determine that the treatment causes a change in the response. So unlike an observational study where we're limited to saying that we just see an association, but we can't make that causal relationship, that strong causal statement, here with an experiment, because we've imposed treatments, we can say that the treatment caused the change in the response. Virtually all scientific research involves conducting a well-designed experiment. Researchers hope the results from these experiments will support a research hypothesis. Example 8. I don't think this should be example 8, but anyway, that's what it's numbered on our page. Salmonella bacteria are widespread in human and animal populations, and there are over 2,000 known serotypes. The reported incidence of salmonella illnesses in humans is about 17 cases per 100,000 people. A food scientist wants to see how withholding feed from pigs prior to slaughter can reduce the size of gastrointestinal tract lacerations during the actual slaughtering process. This is an important issue since pigs infected with salmonella may contaminate the food supply through these lacerations, among other routes including fecal matter and meat juices. He chose 45 pigs from 3 farms. Let's practice identifying the individuals, the population, and the sample. Remember that the individuals are what you're applying treatments to. So here, it's not humans, it's pigs. And what's our population? What do we want to actually say something about? What's the larger group that we actually want to make a statement about? Our population would be all market-bound pigs. And our sample is the smaller group from the population that we actually make observations from. Here, we had the 45 pigs from the 3 different farms as our sample. We've got 3 treatments here. Remember, this is an example of an experiment. So we're going to have treatments. Treatment 1 was no food withheld prior to transport, so let them eat regularly. Treatment 2 is food withheld 12 hours prior to transport. And treatment 3 is food withheld 24 hours prior to transport. Now, a sticking point with some students is that treatment 1. Why do we call that a treatment if there actually is no treatment being applied? Well, it is a treatment group. We have to assign some pigs to that group. So you can think of it as, for instance, level 0 of the dose. So if it's a dosage of a drug, you might want to give one group a placebo. That would be 0 milligrams. So that treatment group, treatment 1, which is doing nothing here, no food withheld prior to transport, is actually considered a treatment. So we've got 3 treatments here. Data were measured on many variables: body temperature prior to slaughter, weight prior to slaughter, treatment assignment, the farm from which each pig originated, number of lacerations recorded, and size of laceration in centimeters. Some questions that we'd want to think about here are, how should we assign pigs to one of the 3 treatments? Well, we've got 45 pigs, we've got 3 farms, and we've got 3 treatments. So it might make sense to go ahead and choose 15 pigs from each farm, right? We want to go ahead and take out the farm effect. So we could make sure that we get some pigs from farm 1, some pigs from farm 2, and some pigs from farm 3. And then within each farm, we need to allocate to the treatments. And so, again, it might make sense to just split it equally. So in farm 1, 5 pigs will be allocated to treatment 1, 5 to treatment 2, and 5 to treatment 3. So here we chose the equal sample size option, and that seems to be the one that most people intuitively go to, and there is actually statistical theory that tells us that's the best way to go in most cases, but for now we'll just go with our gut and write down that we're going to take 5 pigs from each farm to each treatment. I think that sums up our little design that we just talked about. And why would we want to use animals from 3 different farms and not just, for instance, one farm? Well, what if the conditions in that one farm were more unhealthy than conditions in other farms, or way more clean than conditions in other farms in general? What we say we're doing is we want to take out the farm effect. So we want to remove any farm effect on our results. And why might body temperature or prior weight be of interest? Well, these could be an indicator of previous health status. Now, we definitely want you to be able to identify from this example. We want you to identify the population and the sample and that sort of thing and individuals. These last 3 questions that we just answered on the page that we're looking at right now were just to make you think. We didn't expect you to really know any of these, but we wanted to kind of let you know that lots of times you take a gut instinct, you check out if it's okay, and if you find out it's okay, you can go with it. So we just wanted to make you think about experiments and questions that we encounter in a real life setting.